How you doing? This is a short video on the bacterial growth curve. And this kind of explains how and to what degree bacteria can reproduce given a certain environment. And we're assuming in this case that the environment's a good one. So this would be fairly in line with what would happen when we inoculate an agar dish with bacteria, or a bacterium, but more likely more than one. So there's basically 4 steps to the bacterial growth curve. The lag phase, the log phase, also known as the exponential growth phase, a stationary phase, and a death phase. And this is basically kind of what happens. So let's just say right here at time 0, note that these are hours. This is the moment that you inoculated the bacteria onto the agar plate. And what's going to happen is they're going to not really reproduce that much the first hour or two. What they're doing is they're going to be kind of sensing whether or not this environment is hospitable for them to divide. There's going to be certain kinds of food sources that are going to diffuse into the cell, certain amounts of water. Water is very important for life, so clearly this cell needs a lot of water to grow, and that's why one of the good ways to preserve meats in the old days was to salt them, create things like jerky, because it's so salty there's not enough water that the bacteria cannot survive. So it's basically figuring out, hey, is this an area where I can start to make copies of myself? And if it is, it's going to then, after an hour or two, and by the way, we can see that this is a clear solution where there's not that much growth. Then the next several hours, we see unbelievable growth, logarithmic exponential growth, literally a doubling. So let's just say like right here we had 1 bacteria, then we have 2, then we have 4, then we have 8, 16, 32, 64, 128, 256, 512. You get the idea. There's little, just uncontrolled almost exponential growth. But then there reaches a point, and especially with bacteria that do not have flagella, that can't escape, they literally start to grow on top of each other like a big pile of zombies. They can't go anywhere, so they start eating the dead, and all the toxins start to build up. So you have this area called the stationary phase, where there's a roughly, notice by the way, this is the overall amount of cells in solution that you'll see here, or on the plate, that for basically about every amount that's dividing, there's about the equal amount that are dying. And so there's stationary because there's no overall net increase in the amount of the population. And then, of course, as time goes on, and by the way, it doesn't have to be 5 hours. It can be a lot longer than that. It depends on the media. But we can see that over time, what's going to happen is that eventually we're going to see a death phase, is that the number of organisms dying from all of the toxins, lack of air, etc., is going to outweigh the amount of the bugs that are dividing and living. And so that's called the death phase. So that generally explains the microbial growth curve.